All right, let's dive deep into the world of cells, like how they build and recycle themselves. That's like a microscopic city, right? We'll be looking at protein synthesis and lysosomal digestion today.
Yeah, it's pretty amazing. Your body's always busy at that level, building, breaking down, repairing. It's all essential for everything, really, like muscle growth, fighting off infections, you name it.
So let's talk about protein synthesis first. Our textbook actually makes a really cool comparison to spider silk.
Oh, that's a good one. Just like a spider spins its web using silk proteins, our cells create all sorts of proteins for tons of different jobs. It's actually pretty similar, the whole process.
So where does it all start? I know DNA is involved somehow.
Absolutely. You can think of DNA as the blueprint, right? With all the instructions for the cell. And the first step in making a protein is transcription. Basically, a section of that DNA gets copied into a messenger molecule called mRNA, like copying a recipe from a huge cookbook.
And this all happens in the nucleus, right? That's the cell's control center.
Exactly. The nucleus is like a vault for the DNA. Then, once the mRNA's got that protein recipe, it heads out to the cytoplasm. That's where all the action happens. It's like the factory floor of the cell.
Okay, so the mRNA is like a courier taking the instructions to the construction site.
Precisely. And the construction sites are the ribosomes. They're like tiny protein-making machines scattered all over the cytoplasm. And this is where translation happens.
Translation. What's being translated?
Well, the mRNA's message, of course, that genetic code for the protein. The ribosome reads the code and then starts putting together a chain of amino acids. Those are the building blocks of proteins.
So it's like the ribosome is reading a blueprint and then building something, brick by brick, but using amino acids instead of bricks.
Exactly. But here's the thing. The ribosome needs a way to get the right amino acid at the right time. That's where tRNA comes in.
tRNA. What's that?
Think of tRNA like a delivery truck. Each one carries a specific amino acid, and it makes sure that amino acid gets added in the right spot, matching the code on the mRNA. It's super precise.
Wow, it's like a perfectly choreographed dance. Every molecule has its own part.
Yeah, it really is. As the ribosome moves along the mRNA, it keeps adding those amino acids, creating this chain. It's called a polypeptide chain, and that's basically the early version of the protein.
That the polypeptide is like the rough draft,
Yeah,
and then it gets refined.
exactly. When the ribosome reaches a stop signal, it lets go of the polypeptide, and then the polypeptide folds into a specific 3D shape. That's what determines what the protein does.
So it's not just the amino acids, it's also how they're arranged. That makes sense, because proteins have so many different jobs to do.
Precisely. It's like a key fitting into a lock. If the shape's not right, it won't work.
That makes perfect sense. And I actually have figure 2.17 from our textbook right here. It shows how all these molecules work together. It's incredibly complex.
Oh, yeah, it's amazing. It's a great example of teamwork at the cellular level. And there's even more to it. The book also mentions polyribosomes. Basically, it's like a bunch of ribosomes all working on the same mRNA at the same time, like a construction crew, speeding things up.
So it's like having multiple teams working from the same blueprint.
Yeah, exactly. And speaking of efficiency, prokaryotic cells, like bacteria, they don't even have a nucleus. So they can do transcription and translation at the same time.
Really? So they're building the protein while the mRNA is still being made. That's crazy. It's like they've optimized the whole process for speed.
Yeah, exactly. Different cells have evolved different strategies. It's pretty fascinating. But no matter the strategy, the goal is always the same: creating those vital proteins that keep everything running.
Okay, so we've talked about building proteins. Now, what about getting rid of the waste? The book talks about lysosomes. They're like the recycling centers of the cell.
Oh, lysosomes are awesome. They're basically these little sacs filled with enzymes that break down all kinds of stuff, like worn out cell parts or stuff the cell has taken in from outside.
So they're like the garbage disposals of the cell.
Exactly. And they have two main ways of doing this, which you can see in figure 2.21.
Oh, two ways. Tell me more.
The first one's called endocytosis. It's like the cell eats something. Imagine like a Pac-Man engulfing something. And then it gets broken down inside a lysosome.
So that's for stuff from outside the cell.
Right. And then there's autophagy. That's more about cleaning up inside the cell.
Autophagy, is that like self-eating?
Exactly. It's how the cell gets rid of its own old parts. Those old parts get tagged and then sent to the lysosome to be broken down and recycled.
Wow, that's pretty amazing. It's like the cell has its own internal composting system.
It really does. And a key part of this is that the inside of the lysosome is acidic.
Why is that important?
Well, those enzymes inside, they work best in an acidic environment. It's like giving them the perfect conditions to do their job.
Makes sense. So it's like fine-tuning the environment for maximum efficiency.
Exactly. But things can go wrong, of course.
What happens then?
Well, the book mentions that if those enzymes don't work properly, it can cause some serious problems,
Hmm.
like diseases like Tay-Sachs. That's where stuff builds up in the brain because it can't be broken down.
Oh, wow. That's a good reminder of how important these tiny little organelles are. It all ties back to our health in the end.
Absolutely. And speaking of important things, let's talk a bit more about those enzymes themselves. They're not just in lysosomes, they're all over the cell.
Right. I remember learning that enzymes are also important for building things, not just breaking them down.
You're right. They're like catalysts. They make reactions happen faster without being used up themselves. They're involved in pretty much everything, like DNA replication, energy
So
production.
they're like little machines keeping the cell running smoothly.
Exactly. And they have a pretty fascinating way of working. Remember that lock and key idea we talked about earlier?
With the protein shape.
Yeah. So each enzyme has a special spot called the active site. It has a very specific shape, and it only fits a certain molecule called the substrate, like a key fitting into a lock.
So each enzyme is made for a specific job.
Exactly. That's how the cell makes sure the right reactions happen in the right place. And when the substrate fits into the active site, something happens. It might get broken down, or new bonds might be formed.
So it's like a specialized tool designed for a particular task.
Exactly. And the shape of that tool is crucial for its function.
It's also incredibly complex. There's so much more to cells than I ever realized.
Absolutely. And we've only just scratched the surface. There are tons of other processes going on, all working together to keep us alive.
Well, this has been an amazing journey so far. I can't wait to see what other secrets we uncover. Wow, I'm still trying to wrap my head around all that. It's incredible how much is happening inside our cells.
Yeah, it really is. And you know, the book mentions a cool experiment. You can actually try it at home. It shows you how enzymes work.
Oh, I love a good experiment. What is it?
It uses potatoes and hydrogen peroxide, the
Mhm.
stuff you use for cleaning cuts.
Yeah.
It shows how an enzyme called catalase, it's affected by pH levels.
Catalase. That rings a bell. Remind me what that one does.
Well, catalase is found in peroxisomes. They're like these compartments in the cell. Similar to lysosomes, but they deal with harmful stuff, like specifically hydrogen peroxide.
Right, right. And hydrogen peroxide, it's bad for cells. That's why it fizzes when you put it on a cut, right? It's
Yeah.
being broken down.
Exactly. That fizzing, that's oxygen being released. Catalase, it speeds up the breakdown of hydrogen peroxide into water and oxygen. Basically, it detoxifies it.
So in this experiment, we're seeing how the pH level changes how well the catalase works.
Yep. The book gives you all the steps, but the basic idea is you have three hydrogen peroxide solutions. One's acidic, one's neutral, and one's a bit basic.
And then you add potato to each one because potatoes have catalase. And then we see what happens.
Exactly. The more active the catalase, the more fizzing you get. So the different pH levels will affect how much fizzing you see.
Ah, I get it. Like that Goldilocks thing, right? Enzymes have that optimal pH where they work best. And this shows us that visually.
Yeah, that's a good way to put it. You might see a lot of fizzing in one, barely any in another, and something in between in the third, depending on how close the pH is to what catalase likes.
That's really cool. I'm definitely trying that one. But beyond this experiment, why is pH so important in general? For our cells, I mean.
Well, remember, enzymes are proteins, and proteins have that specific 3D shape, right? If the pH is off, too acidic or too basic, it can mess up that shape. It's called denaturing. And then the enzyme won't work right.
So the pH has to be just right so the enzymes can do their thing. It's like a master control for the whole environment.
Exactly. It's all about balance.
Okay, so shifting gears a bit, the book also talks about how lysosomes are made. It even breaks it down step by step, like how it's formed and does its job.
It's like seeing behind the scenes, right? And it all starts with the DNA in the nucleus.
Right, so the genes for those lysosome enzymes, the ones that do the breaking down, they get transcribed into mRNA, just like we talked about with protein synthesis.
Precisely. But it doesn't stop there. The mRNA leaves the nucleus and goes out to the cytoplasm and meets up with the ribosomes.
And then the ribosomes, they build the enzymes, right, using the instructions from the mRNA.
Yep. But those new enzymes, they need some finishing touches. They need to be folded the right way.
So where do they go for that? Is there like a spa for enzymes?
Huh. Something like that. They go to the endoplasmic reticulum, the ER. It's like a network of membranes, a workshop for the cell. That's where the enzymes get folded and modified to get their perfect shape.
So it's like a quality control step, making sure everything's perfect.
Exactly. But they're not done yet. From the ER, they go to the Golgi apparatus.
Ah, the Golgi. That's like the post office of the cell, right? It sorts and packages stuff.
Exactly. The Golgi takes those enzymes, puts them in these little bubbles called vesicles, and then labels them, like, to the lysosome.
Okay, so now we have these lysosomes all ready to go,
Yeah.
full of enzymes. But how do they actually get to the stuff that needs to be broken down?
Well, remember those two pathways we talked about? Endocytosis and autophagy.
Right, endocytosis, that's when the cell takes in stuff from outside, like Pac-Man, gobbling things up. And then it gets delivered to the lysosome.
Exactly. And autophagy, that's for cleaning up inside the cell.
It's like the cell puts a tag on the old parts saying, take this to the recycling center.
Huh, yeah. And then those old parts get enclosed in a vesicle and sent to the lysosome.
It's incredible how organized it all is, both for bringing stuff in and for recycling, like a perfectly choreographed dance.
It's pretty amazing. It really shows how well everything works together. All these different parts of the cell cooperating.
Like a well-oiled machine. And this is happening in every single cell in our bodies, trillions of times over.
It's mind-blowing when you think about it. It really shows the power and elegance of nature. We've learned so much already about protein synthesis, lysosomes, enzymes.
Yeah, it's been quite a journey. I can't wait to see what else we discovered in the last part. Welcome back. It's amazing to think about all those tiny processes happening inside us right now. Building proteins, recycling waste.
I know. It really makes you appreciate the complexity of life. And there's one more thing the book really emphasizes. It's how important the shape of a protein is. For its function, I mean.
Oh, right. We talked about that with enzymes, right?
Yeah.
How their active site has to have that perfect shape, like a lock and key.
Exactly. And that shape comes from the way the protein is folded. It's like origami almost. All those folds create the final form.
So if a protein doesn't fold right, it can't do its job. Like the key won't fit the lock anymore.
Exactly. It's crucial. Even a tiny mistake in the folding can mess up the whole protein. Like an enzyme might become totally inactive.
Wow, that's incredible. Every little twist and turn matters. It's like a delicate dance.
It really is. And scientists are still trying to figure out all the details of how proteins fold. It's a super complicated process, lots of different forces and interactions involved, and things like temperature and pH can affect it too.
Speaking of which, the book mentions mitochondria and how they provide the energy for all this.
Ah, yes. The mitochondria, they're the powerhouses of the cell. They make ATP, which is like the energy currency. All those processes we talked about, they need ATP.
So it's like the mitochondria are providing the electricity for the whole cell to keep everything running.
Yeah, that's a good way to put it. Without mitochondria, the cell would be out of energy.
It's amazing how everything's connected.
It really is. It's all about teamwork. And the book actually leaves us with a little challenge, a thought experiment, you could say.
Oh, a challenge. I'm intrigued. What is it?
It asks us to imagine the journey of a protein, like one that's going to be used in a lysosome, from the very beginning to its final destination.
So we have to trace its path through the cell, starting with the DNA, then the ribosomes, then the ER and Golgi, and finally ending up in the lysosome.
Yep. It's like mapping out a relay race. Each organelle passes the protein along to the next one.
Wow, that's quite a journey. It really shows you how coordinated everything is.
It does, and how important each step is. If anything goes wrong along the way, it can cause problems.
This whole deep dive has been incredible. It's made me realize how much is happening inside us all the time. It's a whole hidden world.
It really is. And it makes you appreciate the complexity and the beauty of life, doesn't it?
Absolutely.
Right.
And it makes you wonder, what else is there to discover? What other secrets are our cells hiding?
That's the best part about science. There's always more to learn, and each new discovery just makes you more curious.
Well said. Well, I think that about wraps up our cellular adventure for today. It's been an amazing journey.
It has. But remember, this is just the beginning. The world of biology is vast and full of wonders, just waiting to be explored.
So keep that curiosity alive, keep asking questions, and keep exploring. Until